My name is Paige Kulling, and I'm a senior in the College of Arts and Sciences, majoring in biological sciences. And I'm an undergraduate researcher in Nelson Harston's lab. I began my research in the spring of 2011. And I originally started on a team with a bunch of undergraduates, and we studied predator-prey interactions and evolution that way. And I really enjoyed that, but I was always really interested in genetics. For graduate school, I planned to study evolution of viruses and bacteria on the genome genetics level. And when I had the opportunity to have an independent research project, I decided to focus on evolutionary genetics. I specifically look at the repair mechanisms that are involved in repairing DNA damage caused by ultraviolet radiation exposure. We use Daphnia because they make really great model organisms. We know their entire genome, and they have phenotypic plasticity, and we know their reactions to different stressors. So we can expose them to ultraviolet radiation and see how a specific gene functions or how it's expressed during that time. We know from previous studies that Daphnia from high ultraviolet radiation ponds repair the DNA a lot faster and more efficiently than Daphnia from low ultraviolet radiation ponds. This would make sense because in nature, if you were getting a lot of ultraviolet radiation, you'd have to repair it better in order to survive. So what I look at is the photorepair gene and how it's expressed. So I want to see if there's a difference in the expression of the photorepair gene between Daphnia from those high UV ponds, which repair their DNA better, and the Daphnia from the low UV ponds, which do not repair their DNA nearly as well. I expose the Daphnia to ultraviolet radiation in the lab, and I also have a treatment where I expose the Daphnia just to visible light, so like what you'd be getting sitting under a lamp. And I then preserve the Daphnia and extract the RNA, and then I turn it back into DNA by a process of reverse transcription. And then I use quantitative PCR, so I amplify their DNA, and I target a specific gene that we know where it's located, and then we measure that repair rate that way using the results of the qPCR. What we found is that Daphnia from high UV environments and Daphnia from low UV environments, they express the same relative amount of photorepair gene. So regardless of how well they repair their DNA, they're expressing the gene that's responsible for repairing their DNA pretty much in the same relative quantities. These results suggest that there could be other mechanisms responsible for the DNA repair rate differences. One could be that there could be a different enzyme encoded by that photorepair gene that could carry out a slightly different function, maybe a more efficient enzyme. Another explanation is that there could be a post-transcriptional step that's responsible for the difference in DNA repair rates. The genetic experiments give a lot of information that the scientific community has not had before, so the research I'm doing is novel, and it can contribute a lot to the scientific community in understanding DNA repair mechanisms, and this could be applied to other organisms as well. This is critical because other organisms are experiencing an increase in ultraviolet radiation with ozone depletion. So it is good for us to know the actual mechanism behind the photorepair pathway and how this DNA will be repaired after being exposed to ultraviolet radiation.